Welcome to the session 2 of this 2-day online workshop. I am Amulya Kotamraju and I'm currently working as a postdoctoral researcher in the Grainful Lab project. I'm happy to chair this session on seaweed cultivation and its fertilizer applications. As a gentle reminder, please submit your questions using the Q&A icon located at the bottom pane of the screen or through the More Options menu, which has 3 dots. Please feel free to post your questions as they come to mind, and I will ask the speaker and seek clarification on your behalf. So let's dive right away into the session. So coming to the first speaker, it is my privilege to introduce the first speaker of this session, Professor Arup Ghosh, a senior principal scientist at the CSIR Central Salt and Marine Chemicals Research Institute in Bhavnagar, and a professor at the Academy of Scientific and Innovative Research. Professor Ghosh currently leads the Applied Phycology and Biotechnology Division at CSIR-CSMCRI. Professor Ghosh's research interests are diverse and impactful, covering areas such as sustainable agricultural technologies, agronomic practices, soil health, and land remediation. He's particularly known for his innovative work in developing seaweed-based biostimulants to enhance crop productivity, improve stress tolerance, and advance the understanding of plant physiology and molecular biology. His research also spans life cycle assessment, microalgae, biofuel, and creating animal feed additives from seaweed to improve animal health and productivity. With over 120 research papers published in esteemed national and international journals, along with numerous book chapters and popular articles, Professor Ghosh is a leading voice in this field. His contributions have been recognized with several prestigious awards, including the Rural Development Award and the Best Technology Transfer Award. He has successfully led over 20 national and international research projects and played a key role in transferring technology to numerous clients. Professor Ghosh is also a fellow of the Crop and Weed Science Society and was awarded the prestigious Raman Research Fellowship. Recently, in 2024, he co-authored a significant policy document titled Strategy for the Development of Seaweed Value Chain, Fostering Diversified Livelihoods, published by Niti Aayog. In addition to his research, Dr. Ghosh is an active member of several important committees in India and has played a crucial role in shaping policies related to seaweed and seaweed biostimulants. He has also convened numerous national and international conferences, delivered over 65 invited lectures, and chaired important sessions worldwide. Today, Dr. Ghosh will share his expertise on enhancing agricultural productivity and quality, the scientific basis and mechanisms of seaweed biostimulants. Professor Ghosh, we are excited to hear your insights and learn from your vast experience. Please feel free to share your screen and begin your presentation. Thanks.
Thank you, Dr. Amulya, for such a nice, detailed biography of mine. I hope I'm audible.
Yes, we can hear you well.
Yeah. I'll just try to share the screen. Can you see the slide?
Yes.
Okay. So, very good afternoon to all of you. And my special thanks to Dr. Arul and other co-organizers for organizing such a beautiful event, and which is being attended by so many people across several nations. Today, my topic of talk will be enhancing agricultural productivity and quality, the scientific basis and mechanisms of seaweed-based biostimulants. Before I do that, I would like to give a brief about what we are. So I am right now a scientist in Central Salt and Marine Chemicals Research Institute, which is one of the 37 Council of Scientific, Industrial and Industrial Research laboratories all across India. And we are in Gujarat. This is Bhavnagar, Gujarat, the one which you can see it's rounded off. We also have a number of extension centers and 5 units. And our ranking among in the world is among top 25 institutions. This is a recent data published in 2023, wherein you can see that we have a good track record of having the highest number of publications, highest number of patents, highest number of human resources developed, PhD per unit of funding what we receive. And we are a government funded research institution, but we also do generate our own private funds by way of different projects or even technology transfers, patenting, and other things. What is our mission and vision? So I'll just tell it in one sentence. It is explore, harness, and transform marine resources for the good of people of India. So what is good for the people of India would also be good for the people of the world. So overall, what we do is we do activities, research activities, R&D, revolving around seawater. So whether it is extraction of salt or whether it is desalination of water, extracting out different marine chemicals, whether it is inorganic, organic or natural products, marine elements. Marine biology, we are specialized in seaweed cultivation. We are specialization in microalgae. We also do cultivation and valorization of halophytes. Then we have marine biotechnology experts, scientists and technical staff who also do marine bioactives and valorization of products for food as well as nutrition. We also do like marine pollution, environment impact assessment studies. We have also done research on microplastics, marine ecology and monitoring and impact assessment. So that is to give you a nutshell of what we do over here in this institute. We are Specialized Marine Chemicals Research Institute, the one and only in India. While doing the R&D, we have collaborated with numerous industries and government organizations, NGOs. You can have a glimpse of these are the people whom we are working with in different aspects, which I just now told. Dr. Amulya just now introduced about a policy document, a very important policy document that calls for strategy for the development of seaweed value chain in India. It was very recently published in June 2024 only, wherein what should be the cultivation strategy, what sort of area do we have where economic cultivation of seaweeds are possible, what are the different value added products what we can do, what should be the strategy of the government. So this strategy document, this policy document encompasses all such things, and we do have contributions. A lot of contribution is from our institute. We are right now also, in fact, doing a mission mode project on seaweeds that has been funded by our parent organization, that is CSIR. Wherein we are also trying to, map different areas across India. As you know, the earlier speakers told that India is having more than 7,500 kilometers of coastal areas which can be gainfully used for seaweed cultivation, and that is exactly what we are doing, other than other aspects of these things. So here is a glimpse of the seaweed cultivation. We do several types of seaweeds. It includes red, green, and brown species. Here you can see Kappaphycus, alvarezii, Gracilaria debilis, Gracilaria edulis, and different methodologies, because I mean, different types of sea conditions will be the determinant of which type of cultivation technology we have. If we have calm waters, we'll use this shallow, I mean, this raft cultivation. But if we have turbulent waters, we have this technique which is called, I mean, tube net technique. So I mean, we also try to develop several different, I mean, cultivation techniques. That's what we specialize in. Very recent development is we have developed, because we have been hearing a lot about I mean, how the seaweeds can be used as animal feed additive. So this was a project that we did few years back. It was a Nimitli project, again funded by our parent organization CSIR, and along with industry partner as well as several other partners of Indian Council of Agriculture Research, which are specialized in animal studies. So we developed seaweed-based feed formulation, feed additives for lactating cows, growing calves, egg layers, as well as broilers. What were the results? We developed several formulations which proved beneficial for improving the performance of both poultry and cattle. We found better immunoresponsiveness, both cellular mediated immunity and HA titer. Then gut health was improved in poultry. Then higher egg production and advancement in egg laying age. So higher calcium and iron content in milk. And most importantly, reduced methane emission and higher energy use efficiency. That is something that we found, and that is very good thing, because India, I mean, India is a big country with a lot of cattle population, and the ruminants actually emit a lot of methane when they breathe in and out. So higher daily growth rate of crossbred calves is another thing which we did. And for many of these formulations, we did toxicological studies also with one of our other sister CSIR laboratory, that is called Indian Institute of Technology, Toxicological Research. So overall, this technology was developed and it was also licensed to the industry. The industry is right now doing marketing trials, and very soon this will be in the market. A very recent development again is this is the last month, I mean July of this year, and we were again granted a project that is from this BIRAC. Okay, that is under Grand Challenges, Grand Opportunities with Bill and Melinda Gates Foundation, and we backed this project. This is again on climate change's impact on agriculture using seaweeds, and this is again about animal feed additives. Okay, so now I come to the plant biostimulants, which is the main topic which I would be covering. You can see that there are several types of seaweeds, but as far as I can tell, in India, I mean, the seaweeds have been used for making biostimulants, I think, I mean, from a long time. But the seaweeds which dominate the world economy are Ascophyllum nodosum, Macrocystis, Eclonia, Durvilia. But in Indian conditions, which we have tropical climate, we do not have any of these seaweeds which are good in, which grows good in temperate conditions. So we have tropical waters, but at the same time, we do have seaweeds in like Kappaphycus, alvarezii, Gracilaria species, Sargassum, Turbinaria. And we have done research on it, and we have found that they also do have, they possess good biostimulant properties. I will be dealing in greater detail in my coming slides. But just for information, the Government of India, Ministry of Agriculture, for which I have also been part of several rounds of negotiations and discussions. So now the seaweed biostimulant, including all the other biostimulant, that they now come under the ambit of fertilizer control order. So earlier the seaweed biostimulants, or as such any of the biostimulants, were not regulated under any of the acts. Now, just like fertilizer, biostimulants have been made a special category, and government has now put it under the ambit of FCO. This was in 2021. So a lot of specifications have now come up, but these are getting refined again and again, because as you know, like biostimulants, it's in its infancy. The regulation is in infancy. Okay, so now how do the temperate seaweed extracts compare with the tropical seaweed extracts? So we did a comparative analysis, and we found that the tropical seaweed species, this one is like for Kappaphycus alvarezii, maybe did the amino acid content and other things as well, and we found that they are comparable, and in some cases they are even much better. So that gave the way to do these studies in a more greater detail, and we did comparative characterization of several other species that are found in India, like Kappaphycus alvarezii, sargassum, YT extract, and we found a lot of total flavonoid content, total phenolics content, ascorbic acid contents, the antioxidant scavenging abilities, different types of polyphenolic species. They were also analyzed, and likewise, and we found that the tropical seaweeds contain a lot of bioactive compounds. Anyway, so what is the final outcome was like, we did the CSIR. We collaborated with 43 state agricultural universities and several institutes of Indian Council of Agricultural Research, and we did the agro trials in several states in multi-crops. So as for example, you can see that we did trials multiple locations, like for example rice, we conducted 38 trials across different states in India, different universities, and we found that when we applied these biostimulants on crops such as rice, maize, green gram, black gram, sesame, soybean, fodder crop, sugarcane, potato, we got yield improvement somewhere in the range of 11 to 37% increase over and above the recommended dose of fertilizers. There were also results wherein we found that we can at least reduce the dose of chemical fertilizer by 25%. Then we, as I told you that 90% of biostimulant still India imports. That is because we do not have sufficient seaweed production, which we are trying to do. That's why we are trying to map several the areas where seaweed can be cultivated profitably and commercially. So we have a huge potential. We can save more than 2,000 crores. Even this 2,000 crores market is underestimated market, and very soon, since this biostimulant sector is now under the ambit of FCO, so we'll get official figures very soon. But overall, after these trials, after the studies, many of the companies, there were licenses. I'm very happy to tell that more than 20 licenses, the technology has already been transferred. And this Sagarika is being, like, the brand name is Sagarika, the product, and it is being marketed by the largest fertilizer cooperative in India, that is called IFCO. So the licensee is now having a turnover of more than 200 crores now. But apart from this, there are other entrepreneurs who have taken technology. For example, you can see the Cripco, that is selling it in the name of Sivarika, and there are several other biostimulant manufacturers who have set up the plant using our technology. And very soon these things will also be in the market. Now, we being purely scientific institutions, but we do take up the industrial research. So we have for this particular biostimulant, we have tried to have a very fundamental understanding on what is the mechanistic action of the seaweed-based biostimulant. So we have published a number of papers in the peer-reviewed SCI journals on any of the aspects which we thought that it may be relevant, whether it is agronomic, whether it is qualitative parameters, how it can decrease the fertilizer requirement, what are the responsive crop stages for maximizing the efficiency, what are the bioactive compounds, what can be the mode of action, what are the roles of the hormones that are present in the biostimulants, what are the other stress buster compounds that are present, for example, quaternary ammonium compounds. We also did a very detailed carbon footprint estimation by life cycle impact assessment at industrial scale. We did it in the industry and we found that it's having extremely low carbon footprint. What can be the effect of these biostimulants on soil properties? What we undertook this as well. What were the effect on soil enzymatic activity, what happens to the soil microbial population. So maybe few of the things I can cover in the next things. We also had a very good understanding of the mechanistic action, the at the gene level, which are the genes which are getting overexpressed, which are the gettings which are getting down regulated, what are the key enzymes in plants responsible for fatty acid synthesis which is getting dysregulated, how it helps in cell wall synthesis, and so on and so forth for both leaves as well as roots, using transcriptomic next genome sequencing approach. And we have also done detailed work on how the abiotic stress can be mitigated by use of this biostimulants on plants. And you can see like a lot of papers have been published. If anybody wants, I can always share. Apart from abiotic stress, we also did work on biotic stress tolerance, and this was also published in several good journals. The earlier example was of Kappaphycus alvarezii, but there are several other red seaweeds and brown seaweeds which are also good, which have good biostimulant properties. So we conducted a very similar, you can see like 166 trials simultaneously we did for the extract of seaweed, Gracilaria edulis, and it was found like again a very good performance and improvement in crops all over India, which we could find. We also did demonstration trials, and we had very good encouraging results. We also did comparative assessment of Gracilaria debilis. That's another species which can be grown in India, for which cultivation technology has been perfected by our institute, and these were also published. A very detailed analysis of what is there inside, which are the hormones that are present, what are the amino acids, what are the types, and not only for plants. There are several compounds what we found they were very important for giving antimicrobial activity or even good for animal health. We also determined what are the physiological concentration. Whether the compounds, the bioactive compounds that are present in seaweeds, they are present in physiological active concentration so as to give activity. For example, literature reports that exogenous application of compounds such as gallic acid at very low levels, like 10 micromolar levels, it improves photosynthetic rates, regulates shikimic acid pathway. So what is the content of this gallic acid in our seaweed extract at application level? Like it is diluted and put as a foliar spray. So our thing contains 11 micromolar. So like this, we have done for several compounds, and we have found these are the bioactives which are present in physiologically active concentrations, which even if one compound is applied, we get evidences of activity, and we have so many together. I will not go into too much detail because of the time. I might have only 5 more minutes left, so I'll be skipping some of the slides, but we do have done elaborate studies on what are the mechanism of action, how the photosynthesis rates increase, how the electron transport rate I mean, it improves how the water use efficiency increases. What are the antioxidant mechanism that is taking place? One good example I can always tell you is like, even though the seaweed biostimulant do not have a lot of nitrogen content, minuscule nitrogen content, but still when it is applied in the plant on the plants, the nitrogen content of the crops improve both in drought as well as irrigated conditions. So what happens? We did, like, we can see that nitrogen content under drought with and without the biostimulant. Sap is nothing but seaweed extract. It increases significantly both under irrigated and drought condition. Similarly, iron and other content also increases in different parts like seeds,
Sorry to
stems, leaves.
interrupt, Dr.
Yeah.
Ghosh. If you could wind up the presentation in the next minute, then we'd have some time to take questions, please.
Sure. We'll be doing it. I'll be doing it. So we did a very detailed transcriptomics analysis of what are the genes that are affected, and we found that the nitrate transporter and the nitrate reductase, another gene that is responsible for converting the nitrate into nitrate, and this is the first rate limiting step that is enhanced. So that's why the nitrate content is increased. We also have found that the application of the biostimulant, this is for red seaweed sargassum, it increases certain overexpression of certain genes, which releases compounds which attracts rhizobium bacteria to the roots, as well as it gives degrades pathogens exoskeleton. Similarly, for soil microbes also, we have found that when it is applied, the soil microbes, which are very important for transformation of nutrients, they also gets increased. I also told about pathogen, pathogen attack. When we inoculated the plants with pathogen and applied the biostimulant, we could find several of the genes like pathogenesis related genes. You can see this PR5, PR3, which gets overexpressed, and as a result, we get tolerance to biotic stress. So this is something which we are now doing in greater detail for different types of seaweed extracts. We also had good results on salinity aspects as well. Somebody was asking what is the carbon footprint. We have done an elaborate study, and we have found that the carbon footprint of biostimulant is extremely low. It's about 118.6 kilogram of carbon dioxide equivalent per kiloliter of the biostimulant production. So when you actually use biostimulants that is having very low carbon footprint for increasing the production while decreasing the high carbon footprint associated chemical fertilizers, ultimately you get more food production per less amount of carbon footprint. So that's something that is having environmental this thing. We do have research gaps still, because a lot of, again, seaweed biostimulant is at a very nascent stage, and we have identified the research gaps, and right now we are working on many of the things like understanding, still deeper understanding, optimization of extraction methods, standardization of products, because still there are no standards, there are no quality assurances. So what are the parameters that has to be seen? And there are different other seaweed species diversity also, which one can look for, because not all the species have been explored well. So these are the things that I'll be doing. So I hope now this is the last what I have to offer within the time. I think I am within the time. So thank you very much for patient listening. Over to Dr. Amulya.